Welcome back, girls. Today we are still going to talk about the types of endosperm. What is the endosperm? It is the nutritive tissue which is found inside the seed for the growing embryo, and it is also a very important part of the human diet. Okay, it's found in mostly in angiosperms, but it's absent in Orchidaceae, Podostimacea, and Trapezae. One thing I have to explain to you that I have not explained is that there are two types of seeds in general. Okay, some seeds where the endosperm is completely absent because the growing embryo has completely devoured the endosperm. Those kinds of seeds are called as X albuminous seeds, like the pea plant. Okay, the pea plant, can you see in the pea plant, you will not find any more endosperm. But in this other plant, the castor plant, because you still have the endosperm still uh, present along with the embryo, therefore it is called as a albuminous seed. Okay, so what did we learn? We learned that there are three different types of endosperm based on the embryo, based on the type of cellular or mode of development. First type is the nuclear endosperm. If you look at the nuclear endosperm, okay, first of all, how is the endosperm formed? In the previous chapter, in the previous video, I had shown to you how is it formed. It is formed when there is double fertilization of the male gamete with the polar nuclei. Okay, the second male gamete will fuse with the polar nuclei to give rise to the PEN. Remember PEN? PEN stands for primary endosperm nucleus. Okay, one of them will fuse with the egg. The first male gamete will fuse with the egg, which is going to form the zygote. And the second male gamete will fuse with the with the polar nuclei to form the PEN. So in this picture, can you see here you have a zygote. How is the zygote formed? By division or by fusion of the male gamete with the egg cell, whereas the PEN is formed when the second male gamete fuses with the polar nuclei. So this is actually inside the embryo sac. So as the zygote is starting to divide, okay, to form the embryo, what happens to the endosperm? The PEN slowly it starts to divide. But the funny thing is that in nuclear endosperm, there is only division of the nucleus. Inside, can you see? There is a nucleus, right? So the nucleus starts to divide. Okay. So here you can see many nucleus, and usually in the cell division you have two methods, two steps, right? You have first step is called as cytokinesis or karyokinesis, which means the division of the nucleus, isn't it? And the second stage is known as cytokinesis. Okay, so what is karyokinesis? Karyokinesis is the division of the nucleus, isn't it? Whereas cytokinesis is the division of the cytoplasm. So the funny thing in not funny, but the interesting thing in nuclear endosperm is you will have only the nucleus which is dividing, but there is no subsequent, immediately there will be no subsequent wall formation yet. There will be no cell wall formation. So it will keep on dividing, giving rise to free nuclei. Can you see these nucleus here, they are called as free nuclei. Why? Because they are not surrounded by any cell wall. So in this type of endosperm, the division of the primary endosperm nucleus and its further divisions are not followed by wall formation. For example, you can find in Limnanthus oxyspora. Girls, one thing I've noticed, this is a important point to consider, okay? Every time I notice while you were submitting your answer scripts, most of you, I don't know if it's not your batch, but most of the girls, they never drew whatever was given along with the notes. So I urge you girls to please take the take out your pens as you are copying down the notes. Please draw the diagrams along with the note, okay? Because diagrams are very, very important. If for 4 marks you get a short note, 3 marks will be based on your writing, and only 1 mark just for the diagram, okay? So it's very, very important. So can you see here, this is another note regarding nuclear endosperm, okay? It's the same note, but I'm just reading it out. In this type of endosperm, the division of the primary endosperm nucleus and a few subsequent nuclear divisions are not accompanied by wall formation. Like I said, there is no wall formation. This results in a condition where the central cell of the embryo sac has formed a few several thousand nuclei which are freely suspended in its sap. That means they are found in the sap. Such a condition of endosperm may persist until it is consumed by the developing embryo. That means you can find this in these 3 important species: Floerkia, Limnanthus, and Oxyspora. Oxyspora, Limnanthus are plants that do not have any cellular wall formation. Okay, but there are certain plants that the nuclear endosperm becomes cellular at a later stage. Okay, what are these plants? Example will be A. califa and phaseolus. In these plants, can you see in this diagram? Here you have the embryo and the PEN or the triploid nucleus, which is formed after double fertilization. After this, you see the PEN divides to give rise to many free nuclei. But after some time, with a short period of time, there will be subsequent cell wall formation. Okay, that kind of nuclear endosperm, this is not the example of cellularization yet, but this is the example of the stages of development of the nuclear type of endosperm. So here you have the vacuole formed in between. But when you talk about cellularization, what does that mean? It means that the plant will start to give walls. It will form walls.